David, welcome.
Thank you.
What we want to talk about is a little bit about what got you into medicine, and then we'll talk some about what got you into research, and what brought you to Singapore, how's it been, and where are you heading?
The whole life story.
The whole life story.
Okay.
Really trying to get an idea, because everyone has a different path by which they come to this almost same thing.
Mm-hmm.
But what drives it? How did it start when you were in wherever, school, college?
So I never imagined in my wildest dreams I would end up the other side of the world doing science and medicine in Singapore. I started back thinking about medicine because my father was a physician, and I didn't have any exposure really to scientists back in my growing up in the suburbs, and then I went to a small liberal arts college where we did science, but there was no such thing as a physician scientist on my radar at the time. But medicine was attractive. It was both intellectual and emotional. You get to talk to patients, and involves a lot of knowledge and thinking. So that was attractive. And so I ended up...
So when you were young, did you actually go with your father to the clinic?
Yeah, I would go on rounds occasionally, go to the office from time to time, become a pest in the office. And then in summers, even in junior high school, I would work in a clinical laboratory, spinning down blood specimens, doing simple clerical work, washing test tubes.
So you were getting exposed to it early on?
Yes.
Okay.
I suppose it's all subliminal. You don't know what you're being prepared for, but you're being prepared.
Right.
And so in junior high school, just simple lab exposure as a clinical laboratory, and then in college I loved science, and I excelled in things like organic chemistry, which other people found more challenging. I just waded right in. And so I was really enjoying doing science and a little bit of research in college, but my college at the time did not have a huge opportunity. But it was enough to whet my appetite. And I think that...
What kind of research were you doing in college?
Oh, it was very simple stuff. It was more planning than doing, because I was one of 7 chemistry majors. It was a very small school. Okay. So we were looking at temperature effects on reductive alkylation using borohydrides.
So one of those famous enzyme reactions?
Even no enzymes, it was all chemical.
Chemical,
so it was solid.
So it wasn't
biological? No, and the biology there was, at the time, biochemistry with that college kind of new, so I didn't have a huge exposure. But then I guess things went well enough that I went to a medical school to Johns Hopkins that really valued research, and so I got caught with the bug, and they offered research opportunities like Duke very early on, and even my first summer I spent in the lab, and decided this was really fun. So I would alternate between doing the clinical work and the lab work, and the clinical work and the lab work, and the school was flexible enough that I could even take off time during medical school to do research similar to the Duke program.
Was there something that started you off at Hopkins in research? Or?
It was just the opportunity that we were, one day the student affairs dean came and said, we have stipends for students who want to do research in the summer. If you find a lab, we'll pay for you. And so I went and looked for a lab. I went with a professor who I thought was working on an interesting area.
Okay.
I think as a student, you don't really appreciate that the faculty have a life outside of the classroom. But the faculty, they may lecture on one thing but do something completely different. So it's hard for the students to know what the faculty are doing. The first lab I picked was really interesting. It was extremely different than what I do now. We were working on, at the time we did not have MRI, we just had NMR. And he wanted to see if you could change signals in NMR by giving certain metals like gadolinium. Mm-hmm. So I was one of the first people to find out that if you gave gadolinium, you could change T1 and T2, and which was this was 1970 something. So that was very cool, and actually it was part of a patent, and so it got my...
And gadolinium is now widely used.
Exactly. So and I was giving it to mice with cancer and showing that it changed T1 and T2 as a first-year medical student. Mm. So it was very exciting.
And it worked.
And it worked.
Okay.
Right. So it's nice to have a project that works. Then the other cautionary tale from that is my mentor failed to get grants renewed, and ended up running a chicken farm outside of Spokane.
Hmm.
So it's...
That's unfortunate.
Yes. So the science is complicated. Life is complicated. But then, so subsequent years, I decided that was interesting, and I would go look for other labs. And I had the great fortune to end up in Van Bennett's lab, because I was interested in hematology. He was interested in red blood cell membranes. He had a fellow in his lab named Peter Augury, who I worked with on a project, and those projects worked well. And I had a paper with Van and Peter in JCI, and so one thing led to another, and I just sort of decided this was a lot of fun. So it's a nice complement to doing medicine. In medicine, you spend a lot of time working on what's the facts on the ground today, and in science, you can say, how do these fit together to, there's gaps here. How do I ask questions to fill those gaps? So it's a nice complement.
And so how did you get into pediatrics?
A mentor. I did, well, there's two parts. One is I like children more than I like adults.
Okay.
When children are sick, you actually really, it tugs at the heartstrings, but you feel like you can do something to help them. And a lot of adult diseases tend to be brought on by lifestyle, and adults get more when adults are oppositional, it's less fun to deal with them.
Okay.
When kids are oppositional, you understand. For me, it worked out well. So I enjoyed working with children. And then I had a mentor, a pediatric hematologist who was senior, who was just extremely committed to the physician-scientist sort of ethos, who was a superb clinician, but also had a research lab, and took me under his wing, and we did a small research project together, but just to see the dedication and the ability to move from clinical work back to basic science and back and forth again.
And that was at Hopkins?
That was at Hopkins.
Okay.
So I stayed. I was there for 13 years because there's a very strong research ethos, and they're extremely supportive of it. And at the end, there was other opportunities.
So you joined as faculty after you completed your training?
Yes.
Okay. And then what happened?
Well, then, so I was finishing up my training, and the true story is that my wife told me that she was going to move to Utah, and I was welcome to join her.
Okay.
So I moved to Utah.
You won that on? You won that on tape. Okay.
We were 13 years in Baltimore, and so I guess one key to a successful career is an incredibly supportive spouse. And so life is always a series of trade-offs and compromises, and I was really enjoying Hopkins. But at the end of the training, she was not interested in staying on the East Coast, and so I got a faculty position, but she urged me to look at other options. And Utah turned out to be a great choice because they just started a program called the Human Molecular Biology and Genetics Program, which was designed to take new physician scientists and put them in a basic science environment. I think one of the failings of physician scientists in clinical departments often is that they're lost, that they are clustered with people who don't do what they do, who don't understand their lifestyle, who don't understand what's important to them. And so Utah had built a building, had some world-class scientists in it, including one who won a Nobel Prize, and we're interspersing them in amongst the basic scientists, we're bringing in a cadre of physician scientists. And Utah is a beautiful place. They pay more than the East Coast schools do. And the lifestyle was nice, and it was an incredibly supportive environment. And I had a mentor there. Again, it's critically important to the career, is my boss was extremely supportive of doing research as a clinician, so that I had 80% of my time protected, and I could go into the lab and I wouldn't get called out, and I could concentrate on it. And the nurses would make fun of you for being a lab rat, but the important thing is your boss doesn't call you in the middle of an experiment and say, I really need you to clinic today because what you're doing isn't as important. And so that kind of support was super. And so that was 17 wonderful years in career development in Utah.
And what did you work on?
So I started continuing a project trying to understand how replication of DNA is controlled. And that led me to, the thing that launched me in Baltimore is discovering a protein phosphatase that controlled DNA replication. And we continued those studies in Utah and expanded our knowledge of these interesting enzymes, and those enzymes also turned out to be involved in the Wnt pathway, and the Wnt pathway had been put into the colon cancer pathway by a discovery at Utah by Ray White's group. And so all of a sudden, instead of working on DNA replication, we got involved in the Wnt pathway. And so that's led to our studies that have continued on in Singapore about how the Wnt pathway is regulated, how these different enzymes regulate it. And for those of you who don't know, the Wnt pathway is really important in the control of cell proliferation, so it's very important in development and in cancer. And so that led to the ability to study it extensively there. And then when we came to Utah, when we moved, the opportunity in Singapore was to have a time frame where I wouldn't have grant pressure, where I could start a new area, and that was to look for drugs that would impinge on the pathway. And it turned out to be a great move because we had Alex Matter in the Experimental Therapeutics Center here, where
so you had the right partner.
Fantastic collaborators, the right partner at the right time, and that's gone. It's gone well. It's always we are pioneers. This is our drug is the first big drug project for Experimental Therapeutics Center. So there's been bumps in the road and things to iron out, but overall it's worked out really well. So we're really pleased. We have a patented compound. It's got effectiveness in mice. We hope to go into humans in the next year or so. That's been very exciting.
So when you were in Utah, were there particular things that happened which made you go in directions that you didn't expect? I mean, the Wnt sounds like was
that
something.
was completely serendipity. So the way my lab got involved in the Wnt pathway was a seminar that a postdoc from Ray White's lab gave, and she had done this screen and identified a number of genes that interacted with this major component in the Wnt pathway. And she was throwing up gene names on the board. Most of them were names like, were Riken 472. Just gene names that didn't mean anything. But we had done a similar screen in our pathway and had come up with the same gene names. And just this pattern recognition, I saw a number on the board that corresponded to a number we had just isolated, and the light bulb went on, and that started it all. So it's just, if there's a message there, it's go to seminars that aren't in your field and listen, because you never know what's going to come up. The ability to take distant points and connect them is I think it's the beauty of being a physician scientist. Is a PhD has great depth in certain areas, but he doesn't have necessarily the breadth of human disease experience. So if someone shows me one finding here, I can relate it to a human disease over here, make a connection.
So becoming a physician scientist really combines both. You get an idea why you're studying it, and you're also getting an idea when you see something, connecting it to something else because of your background.
Mm-hmm.
So what would you tell a young person starting medicine? Why become a physician scientist? It's not the easiest road.
No. It's not the easiest road. It's not the best paid road. The hours are just as long as clinical medicine. The rewards are very different. So you don't have, when in patient care, there are some really wonderful rewards. The patients love you. The nurses love you. You make people better every day. You go in, there's a problem, you deal with it, you move on to the next problem. So every day you have short-term solutions and short-term positive reinforcement. When you go into science, there are long periods where things don't work, where the experiment's not working, the idea was a good one but turned out not to be correct, and you have to be willing to cut your losses and move on to other areas. But the fun part of it is that you get to go places no one has ever gone before. You discover things that no one's ever seen before. You make a contribution that's acknowledged by your peers as a major insight that helps you solve problems. And over the space of the last 20 or 30 years, the progress has been just amazing. The development of all these targeted therapies, because building on these little steps that scientists make, we can then start to apply this to clinical medicine in really productive ways.
So really being curious and exploring.
If you're curious, if you like to say, I don't believe this is true and I want to get to the bottom of it, you have to be willing to question accepted wisdom, because accepted wisdom is usually a house of cards that we build. And you have to be willing to put up with ups and downs, but the ups are very good, and the downs are livable. but you have to be curious and dedicated and enjoy getting your hands dirty and pipetting all day and going back and going over the numbers and looking for patterns and trying to solve puzzles.
And when you talk about it, your mentors played a big role.
Absolutely critical.
So what do you look for in a mentor in retrospect? I assume you've had good mentors and not
Yeah,
some
if they're not
so
good mentors,
ones.
they don't end up getting called mentors. So it was never something I did consciously, although I think nowadays we're much more conscious of its value. But the way I would choose a mentor is I went and talked to people about potential projects and decided whether or not there was good chemistry between them and me, that they were interested in the project, that they were interested in me personally, that they were the kind of people I would like to be when I grew up. And
did that work, finding people that way?
absolutely. Well, it worked well for me.
Worked well for you.
I mean, I some of this is total luck. This is a path that I would never have predicted. I just, kid from the suburbs going to medical school, and I thought, probably I'd end up, practicing medicine. The ability to connect with people at a research-oriented medical school who are passionate about science and passionate about research was able to sort of take whatever glowing embers I had of interest and fan it into a small flame. So it's being in the right environment, having the interest first, being in an environment that nurtures it, and finding faculty members who think this is a cool thing to do.
And having the time, the protection.
Absolutely critical, because you can be as excited as you want, but it can be beaten out of you by the beeper going off one too many times. There's nothing quite like a beeper going off to ruin an experiment.
So moving to Utah ended up being really very, very good for you.
Absolutely.
So what about
Fresh
coming to
start.
Singapore? What made you look at Singapore?
So I was in Utah for 17 years and had risen to a point where I was interested in doing more than just continuing on as I mean, I was enjoying the research, but was interested in a little bit more leadership opportunities. And I mean, it never occurred to me to move overseas, but the true story of how I got to Singapore is I saw an ad in Science about Duke in Singapore, and I saw Pat Casey's name on it. And I knew Pat. Pat and I were friends from meetings in the U.S. And I didn't know he had moved to Singapore. But so the most important first step was I asked my wife, "What do you think?" And she said, "Okay," because she's very adventurous also. And so I emailed Pat, and one thing led to another. And what attracted me to it was, well, it's a startup, right? How often do you get to do a medical school startup? It was clear that Duke is a first-rate operation, and there was going to be the ability to do solid science. I'm very committed to the idea of training physician scientists, which the school is. And I get the chance to see a part of the world I never thought I'd see. So I get to bring the kind of a Western perspective, and Asia has been very educational in terms of...
So was this the first time for you to come to Asia, or have you been in Asia before?
I had been as a tourist a couple
Okay.
of times. But certainly being a tourist and being taken care of on a tour is very different than having to open a bank account, get a cell phone, figure out how to get cable service to your house, buy furniture.
It's really a brand new start.
Brand new start in a
And a
culture
big
that's
contrast
similar
from
but
Utah,
not
right?
different. A big contrast from Utah. Well, I'm still a minority.
Okay.
Utah is 80% Mormon, 70% Mormon, and Singapore is so I'm a minority here as well.
So, and the weather is clearly different.
Yeah, I guess if there was one thing I could change about Singapore, it would be
The weather.
to tone down the humidity a little.
Okay. So after you came here, how's it been? You've been building a program. It was a different thing to find new people, get people together, and build a program. How's it been? How's that whole
It's
aspect of your life been?
busy.
That's good.
It's never boring here. So recruiting was fun because I had no idea what to expect. We started recruiting junior faculty to fill up the program in cancer and stem cell biology. And we put an ad in the paper and sent around letters to various departments, and we were really happy with the quality and the number of applicants because you had no idea whether people are going to be interested in moving. But it's a Duke-style recruitment package with a internationally competitive salary and internationally competitive startup funds. And the kind of people we attracted were people of Asian ethnic background who had been training either in the U.S. or Europe. So it had various motives. They weren't uncomfortable moving to Asia. They were closer to family, but this was probably a high point in what we hope is a high point in research in Asia. And so they had the opportunity to maintain a high level of science and be back in Asia. So we had I've been very pleased with the quality of the applicants. I guess it's interesting to note that we didn't get many kind of Caucasian Americans applying to a Duke branch out here. So I've learned that maybe I have more of the thrill-seeking gene than most.
I think you need to have a sense of adventure if you want to make a big move.
It is. The challenges are that if your family is all in the U.S., you're a long way away from your parents and your siblings, and your children have to change schools, and so there's a lot of changes to be made. So it's more challenging than moving to another state in the U.S., for instance, or even moving to another country in Europe.
So you've also had students work with you.
Yes.
How's that been over the years, not just Singapore?
Well, students are what keep you young. Because students ask questions that they don't know they shouldn't ask. Questions that are, in a sense, naive, but pull together questions from a much broader perspective. I think after a while if you think about an area for a while, you tend to get into a little bit of a rut, and students aren't in that rut, so they're asking a question that is very broad. And so it helps jerk you out of your rut a little bit when you try to answer it. And it's fun to go through the primary data with the student and to try and piece it together and then try and help them see where there's patterns and where there's things that don't fit the mold. And the biggest challenge for students everywhere is to realize that just because the textbook says it's so doesn't mean it's so. And if your data doesn't agree with the textbook, there's two possibilities here and you have to consider them both. One's the textbook's wrong, the other is the textbook may be right, and your data is somehow flawed, or your data is absolutely right and the textbook's flawed. And I think it comes out pretty even. The textbook is based on the best guess at the time.
And it really allows you to explore. That's the main thing.
Yeah.
So going forward with your own research, where do you see it going?
So we're very excited about the drug development because this is a first for me, and as a physician scientist, I think that one of the exciting opportunities is to actually you want to make a difference in people's lives. And so coming up with a relatively novel approach to drugging diseases and being able to see a drug go from an idea using a high throughput medical student to a screen with cutting edge robots to superb chemistry over at ETC, and now seeing it actually work in a mouse is very exciting. So now the goal is to figure out who to give it to on a molecular basis, to come up with biomarkers, to get it into patients, to predict and hopefully mitigate side effects and see if it has efficacy. And then we learn things. Every time you get a new drug, you get a new tool. So there are things we learn from giving the drug that we didn't expect, and that offers new avenues of research.
So it allows you to explore more.
Right. We're only as good as our tools, and the drug is an exciting new tool for us.
And what about the program?
So the program is, 5, 6 years, I guess 6 years now. Some of our faculty have been here 4 to 6 years and are doing really well, on track. They're getting grants, they're publishing papers. We have a lot of graduate students who've joined the program, some really good postdocs, both locally and from around the world. So it's just flourishing. I mean, I think at steady state, a program is a well-maintained garden, and it requires a constant attention every year, but it's being very productive.
So we have a lot of students who come in, and they all have to do research in the third year.
They all get to do research in the third year.
All get to do research in the third year. How do you attract them to your program? Or what can you tell them that'll make them think about joining your program?
it's the native interest, the basic interest of the student has to be, they have to think that basic science is exciting and a career path for them. Because, I mean, as I started off in this, basic science was obviously a career path because I was doing it in the laboratory of other people who were doing it. And I think students need to see that there are successful physician scientists who have combined clinical work and research as their career path. And if they're interested in basic mechanisms, this is the best opportunity they're ever going to get. They're never going to get the opportunity again to spend a year learning cutting-edge methods, basic molecular mechanisms, high-tech machinery. They'll always have the opportunity to see patients and do clinical research, but they'll never again have the opportunity to immerse themselves in this kind of very basic environment where they can get at the very roots of diseases and understand mechanisms.
So David, thank you.
It's been a pleasure.
Anything else you want to tell a potential audience, which is going to be hopefully a lot of students?
You never know where the path is going to take you. You just, go with what seems like a good idea at the time, take some chances, realize that, having an MD is a little bit of a safety net. Even if you do the science route and it doesn't turn out to be the right path for you, there's a fallback position. So you can take more risks than you would otherwise take, and just go for it.
Thank you.